before a single breath is taken, before the heartbeat steadies into rhythm, before organs rise from clusters of cells, another structure forms in silence, an organ unlike any other in the human body, an organ built from two individuals at once, shared but separate, fused yet distinct. The placenta. It is the only temporary organ humans create, an organ that forms in mere weeks, sustains life for months, and then disappears the moment its mission is complete. It keeps two blood systems apart yet connected. It nourishes, filters, communicates, and protects with a precision so refined that the fetus survives in an environment that would otherwise be impossible. Most people imagine pregnancy as the story of the growing fetus, but the deeper truth is this: the placenta is the unseen architect, the engineer constructing a living environment where oxygen, nutrients, hormones, and immune signals are exchanged with microscopic accuracy. How does an organ form so quickly? How does it fuse with maternal tissue without triggering rejection? How does it deliver oxygen without mixing blood? And how does it decide what enters the womb and what stays out? Imagine an organ acting as a lung, delivering oxygen, a liver processing nutrients, a kidney filtering waste, an endocrine gland shaping hormones, and a security system scanning every molecule, performing all these roles at once. Imagine it building itself cell by cell, reaching into the uterine wall, forming a biological bridge between two separate systems. This is the placenta, one of the most extraordinary structures the human body ever creates, yet one most people never truly see. To understand pregnancy, we must step into this hidden world, where two bodies collaborate to build a third. Let's go inside. Inside the womb, the placenta forms with a level of engineering that feels almost intentional. Cell by cell, signal by signal, it builds a living interface between two bodies that must cooperate without ever fully merging. To understand this process, we need to zoom in to the early days of development, when the embryo is still a microscopic traveler and the uterus is preparing to receive it. Once fertilization occurs and the embryo develops into a blastocyst, two main populations of cells emerge. The inner cell mass becomes the fetus. The trophoblast becomes the placenta. This distinction is not cosmetic. It's architectural. The trophoblast is already specialized for invasion, communication, and nutrient extraction. These cells are the first to sense the uterine environment, the first to engage with maternal tissue, and the first to transform the endometrium into a space capable of supporting gestation. When the embryo reaches the uterus, the trophoblast cells begin their controlled invasion. They differentiate into two essential layers. The inner layer, called the cytotrophoblast, remains individual and organized, providing structural stability. The outer layer, the syncytiotrophoblast, fuses into a continuous multinucleated sheet that spreads across the uterine lining. This fused layer is extraordinary. It behaves like one giant cell, secreting enzymes to gently dissolve maternal tissue, forming small spaces called lacunae, where maternal blood begins to flow. This is the earliest blueprint of placental exchange. As development continues, finger-like projections, chorionic villi, extend into the blood-filled lacunae. These villi are the functional units of the placenta. They branch like trees suspended in a lake of maternal blood, increasing surface area and creating the microarchitecture that allows the fetus to receive oxygen and nutrients without direct blood mixing. Here, fetal capillaries develop inside the villi, while maternal blood surrounds them, separated by just a few layers of cells. It is an elegant compromise, close enough for exchange, distant enough for safety. Meanwhile, the embryo forms its own contribution, the umbilical cord. Inside it run 2 arteries and 1 vein, linking fetal circulation to the placenta. But these systems stay separate. The fetus sends deoxygenated blood to the placenta through its arteries, receives oxygen-rich blood back through the vein, and never directly touches maternal blood. This separation is essential. Mixing the 2 systems could trigger massive immune reactions. Instead, diffusion, transport proteins, and concentration gradients make the exchange possible. By around 12 weeks, the placenta is fully established. Its metabolic workload is immense. Every minute, maternal blood floods the intervillous space. Every minute, the fetal side absorbs oxygen, glucose, amino acids, and micronutrients through specialized transporters. And every minute, carbon dioxide, urea, and waste are returned to the maternal circulation for disposal. In this sense, the placenta replaces the baby's future lungs, liver, and kidneys long before they exist. But its responsibilities extend far beyond exchange. The placenta is also a powerful endocrine organ. It secretes human chorionic gonadotropin, HCG, the hormone that keeps progesterone levels high and signals to the body that pregnancy has begun. It produces progesterone itself, sustaining the uterine lining and preventing contractions. It synthesizes estrogen, shaping blood flow, tissue growth, and development. Human placental lactogen modifies the mother's glucose and fat metabolism, ensuring a steady stream of energy to the fetus. These hormonal adjustments reshape the entire maternal physiology. The placenta also acts as a selective shield. Its structure filters harmful substances while allowing protective immunoglobulin G, IgG, antibodies to cross, giving the fetus early immune protection. At the same time, specialized immune cells within the placenta create a local environment of tolerance, preventing maternal rejection of the genetically distinct fetus. This immune microenvironment is incredibly nuanced, balancing defense with acceptance, protection with coexistence. Communication between placenta and fetus is continuous. Hormones, growth factors, and molecular signals regulate fetal development, influence how nutrients are allocated, and adjust placental behavior based on fetal needs. If oxygen levels drop, the placenta increases blood flow. If nutrients become scarce, it modifies transporter activity. It is not a passive organ. It is an active negotiator. Throughout pregnancy, the placenta grows, remodels, and adapts. Its villi increase in complexity. Its blood flow rises dramatically, and its endocrine signals evolve as the fetus matures. By the 3rd trimester, it is performing at peak capacity, handling the metabolic and respiratory needs of a rapidly growing human. But this entire system, the architecture, the signaling, the hormonal shifts, remains invisible, unfolding in a darkness where 2 bodies collaborate without ever merging. And as birth approaches, the placenta prepares for its final act: detachment. The placenta is not simply a vessel or a nutrient pipeline. It is a dynamic organ of negotiation, protection, communication, and transformation. Its story is inseparable from pregnancy itself. The placenta's story ends the same way it begins: in silence, in cooperation, in the space between 2 bodies that learned to work as one. It is easy to think of it as simply a temporary organ, a supporting structure, a byproduct of pregnancy. But when we look closely, the placenta becomes something far more profound: the first environment a human ever inhabits, the first bridge between self and other, the first negotiation between protection and growth. Throughout pregnancy, the placenta performs a choreography of impossible complexity. It filters without mixing. It nourishes without overwhelming. It shields without isolating. It balances two immune systems that should, by default, reject one another, yet instead learn to coexist for months. It adapts to fetal needs minute by minute, adjusting oxygen flow, redistributing nutrients, amplifying or quieting hormonal signals. It is not just supporting life, it is shaping it. The placenta reminds us that development does not happen in isolation. Every heartbeat inside the womb is powered by its architecture. Every breath a newborn takes is the first moment of independence after months of placental respiration. Every shift in maternal physiology, from metabolism to immunity, is guided, in part, by placental signals that ensure survival. And when its mission is complete, the placenta steps aside with astonishing finality. It detaches, exits, and disappears. An organ built for one pregnancy, one story, one life. No other human organ is grown, used, and released with such purpose. In that sense, the placenta is a lesson in biological grace. It teaches that connection is not weakness, but strategy. That cooperation is not compromise, but design. That life begins in relationship, from embryo to uterine wall, from villi to maternal blood, from two systems learning to support a third. A temporary organ, an irreplaceable bridge, a silent architect of existence. The placenta forms in darkness, works without pause, protects without recognition, and completes its purpose the moment a new life begins.